Okay, so to really understand this whole thing with sialic acid and what it's doing on proteins, I think we've got to go into a little bit of what glycosylation is. And so this is also stuff that's really useful in any sort of protein biology. It comes up in cell biology all the time. So it's worth understanding if you haven't already got a good picture of it. And if you got a good picture, we'll try and make that a little more detailed picture. Why not? Yeah. Okay, so there's two big kinds of glycosylation in the world. One is called N-linked, and the other one is called O-linked. N-linked is linked at a nitrogen. Neat. And the nitrogen is in an asparagine amino acid, and the single letter amino acid code for asparagine is also N. So it is N-linked, and it's linked to an N of N. Neat. Um, the O-linked glycosylation is linked to one of two amino acids, so either serine or threonine. And so their single letter codes are S or T, just like it says there. Serine and threonine both end in a hydroxyl group, which is an OH, and it's the O right there where we're going to attach the sugar. So you can see on the left and the right at the bottom in all the colors, these are the amino acids. And so we're showing them as kind of a simplified form with their side chains pointing up and the main backbone running along the bottom from left to right. Attached to the side chains, you can see the sugar molecules in black, and this will be the first sugar that is added. And so the first sugar that gets added is usually a thing called N-acetylglucosamine, and you can see it on there. Um, N-acetylglucosamine is something that people sell in little capsules with the idea that eating it will stop inflammation or protect your joints. I don't believe it actually does either one of those things, but there's no harm to it. All it is eating a sugar molecule, and it gets broken down into atoms, and then we absorb the atoms, and then that's fine. It works as a food. It just doesn't have this other mysterious property that it's supposed to have. So the places in proteins where you can link one of these things are called glycosylation sites. And so they have a specific structure. And the specific structure is going to be three amino acids. So for an N-linked glycosylation, the first amino acid will always be asparagine, or N. The second one can be whatever you want it to be. So that's why there's an X there. X means any amino acid, unknown amino acid. And the third amino acid has to be either a serine or a threonine. So NX and then S, or NX and then T. That is a N-linked glycosylation site. And if you have those amino acids in that order and it's on the surface of a protein, and the protein is made with that part poking into the endoplasmic reticulum, then it will be glycosylated. And we'll add a big long chain to the end of this. I'll show you the chain here in just a minute. It's pretty cool. O-linked glycosylation has a simpler signal. It is just a serine or threonine with no asparagine there, but it also does have to be on the surface. O-linked's a little less common than the N-linked, but yeah, you gotta have just the right combination of sequence and structure for an O-glycan, and we're not sure beyond the serine or threonine what you need. Okay, so shown here, these are some proteins, and these are some of the proteins that add on the glycosylation signals. So I just thought maybe you'd like to see what these things look like. And so this is the structure of some of these proteins. Yeah, it's kind of neat. So that's all the little colorful drawings up there. I don't know that we need to do anything more than that with it, but each of these things will pick up the sugar molecules and it will start gluing them on. It'll start making a little chain out of them. And you can see one of the chains on the next slide. Let's see this chain. Okay. So these are examples of some of these chains rather. You'll start out with the blue thing, which is an N-acetylglucosamine, so glucnac. That's how we abbreviate that. Glucosamine N-acetyl, glucnac. The other things that we can put on are shown there. So we're going to have a neuraminic, the NUAC. You'll have the galactose, galnac, and you'll have regular just galactose and glucose and mannose sugars, um, and fucose as well. And so each of these things is attached at a specific position by a specific enzyme, and they form big long chains. So you notice how all these chains that are here, they look like a Y, but they end in a little letter X. The X means that it goes on, and from this point, it can keep branching or it can keep elongating. It can be, 20, 30 of these sugars long if it needs to be. But yeah, that's how it's going to form. And then what they're not showing you is that if you get to the very edge of it, to the very top, um, we're going to sort of trim these back a little bit, and then we put a cap on the end of them so they can't grow anymore. So we can't just glycosylate forever and make a giant unwieldy mass of wasteful sugar. And it is going to be that neurominic acid, which is also called sialic acid. So they're the same thing. Sialic acid is neurominic acid. So neuraminidase was named after neurominic acid, which is sialic acid. So another word for neuraminidase is you can call it a sialidase, which means the thing that cuts sialic acid. And to a scientist that knows what they're talking about, those two things mean exactly the same thing. We say neuraminidase because that's just the one that hemagglutinin is called, like the H1N1. That's an N for neuraminidase. So that's just yeah, that's just how we read them. All right, and so then the last thing in here is if you wanted to see what the glycosylation structure is, you can put in an enzyme called PNGase, and PNGase type F will go in and it'll actually cut off the glycans. So on each of these pictures, you can see the ASN is another name for asparagine. So that's the amino acid with an N that has a nitrogen in it. So this is N-linked glycosylation. And when you put in the PNGase, it will cut off those structures unless there is, for example, a fucose at the alpha 1,3 position at the bottom. And there you see the little X over the arrow. So it's not cutting that one, for example. I think this is going to come up a little later in one of the slides. But there are two ways to attach. Alpha, yeah, no, that's two ways to attach fucose. There's two ways to attach sialic acid, and those are alpha 2,3 and alpha 2,6. But we'll get to those more in a minute. Thank you.